Okay, well, good evening, everybody. It's a great pleasure to be able to introduce this inaugural lecture this evening, and particularly like to welcome Keele colleagues, also other academic and research colleagues of Frederick, and in particular extend a welcome to Frederick's family and siblings that are here. It's really great to welcome you today. Just say a little bit about the role of inaugural lectures, and I think they are quite tricky, I think, and not putting any pressure on Frederick, but in the sense that they are a celebration of the professor's area of expertise, and they also help raise the profile of research at Keele and in the discipline, in this case life sciences. And I think the bit that's really tricky about them is that amongst the audience there are some very expert specialist colleagues, and then there's people like me who have probably pretty limited understanding of Frederick's research area. And Frederick's research area is in the area of behavioural ecology and malaria, so it's obviously a really key global societal challenge, so really an example of very high impact research that is undertaken at Keele. So I'm just going to give a brief biography of Frederick. And so Frederick undertook a BSc in biology at Lausanne University in Switzerland, followed by a PhD in behavioural and evolutionary ecology of host parasite interactions at Bern University, also in Switzerland, before moving to the US to undertake his postdoctoral research. He spent a total of 6 years in the US at UCLA, where he did research studying, focusing on social insects, and then at University of Texas Medical Branch and Davis University, also part of University of California. He was then appointed to lectureship here at Keele in the School of Life Sciences in January 2006, so just over 10 years ago. And he was promoted to a readership in 2011 in recognition of his research achievements. And then in this year, he was then promoted to professor, again in recognition of continued research success in his research area. And Frederick's professorial title of Professor of Medical and Molecular Entomology highlights the area that Frederick has really made very significant research contributions. And at Keele we have two sorts of professors. We have those that are appointed externally and those that have been promoted from within in recognition of their research achievements. And in many ways it's often more difficult to get promoted internally than it is to be promoted directly to a professor post. So Frederick is, say, the example of someone that's been promoted internally, having been appointed here originally as a lecturer. Just going to very briefly go through a couple of achievements in Frederick's research career since his appointment to lectureship at Keele. So he's a very active member of the what we call CAPE, the Centre for Applied Entomology and Parasitology, and published a large number of papers in high impact factor journals, which is what one would expect for anyone promoted to professor at Keele. And his research is genuinely global, and and the fact that it is around tropical diseases, malaria, has led to invitations literally throughout the world to present his research. In 2015, he was made a Fellow of the Royal Society of Biology and also a permanent member on the Wellcome Trust Public Health and Tropical Medicine Interview Committee, which, again, recognition of the esteem in which he's held. Going back to 2007, not long after Frederick had been appointed at Keele, he set out plans for large collaborative training grant with colleagues, both from Keele but also from Mali in Africa. And that proposal, with its strong African capacity building component, led to an £800,000 Research Council award, so a very significant research award to look at genetic engineering in mosquito vectors for the control of malaria treatment. And in turn, the success of that research and the research track record that had come from that work, as well as the innovative ideas, led to a partnership with leading research institute in Burkina Faso, also in Africa, where secured £1 million research funding for project targeting male mosquito mating behaviour for vector control. And again, this is a really significant research project employing not just a good number of staff, but having really significant impact on research capacity building in Africa, and has led to some really important breakthroughs in mosquito ecology and control that will contribute to novel integrated malaria control programs. I think one of the things that I think Frederick will bring out in his lecture is that these capacity building grants, as well as undertaking really high quality research, have involved design and supervision of major construction projects, construction works, and they've received a lot of media attention and really played a key role in training future generations of scientists and hosting international collaborative projects in Africa, as well as significantly enhancing Kiel's international research profile. And again, an example of the esteem that Frederick's research is held in is that he acted as a consultant on a large consortium grant that was sponsored through the Bill and Melinda Gates Foundation, focusing on mosquito genetic control programs. I think it's also important to emphasise that vast majority of academics at Kiel are involved in both research excellence but also in academic teaching delivery, and Frederick is director of our MSc programme in molecular parasitology and vector biology, and has been for the past 5 years, so a direct link between the leading research that he undertakes and our teaching. So finally, I'd like to formally welcome Prof Tripet, Professor of Medical and Molecular Entomology, invite him to give his inaugural lecture of mosquitoes, rice and man in a time of prospective malaria eradication. Frederick.
Thank you, Mark, for these kind words. And welcome to everybody who showed up today. And I'm very happy to see you, especially the family and those people who traveled from very far away to see this summary of some of the research I've been doing in the last 20 years, focusing on mosquitoes and malaria. I would like to start this lecture and journey by going back to one of those crossing points or turning points in my research career. In '98, I was based in Los Angeles at UCLA, and I was finishing a fellowship focusing on social insects. It was an interesting topic, but it was fundamental research, and we were looking at the genetic determinants of aggression in ants. When the fellowship was finishing, I was due to go to the Max Planck Institute in northern Germany, and at some point I decided, okay, I will give America another chance. I canceled the German plan, and I was looking for a job in Los Angeles. And fortunately, within 2 weeks, a population geneticist at UCLA came to talk to me, and they said, we are looking for somebody willing to travel to Africa who can speak French in English and can troubleshoot problems. Okay, I said, that sounds great, and it's on mosquitoes and malaria, and I thought, well, this is an opportunity for finally my family to understand I'm doing something useful, and I'm going to switch to something more applied, use the same tools, but switch to an applied topic. And here I go. Within 3 weeks, I packed the few things I had, and I headed for Galveston, Texas, which is a very hot island on a sandbar off of Houston. And it's very muggy. It has got a lot of mosquitoes. And there, within 2 weeks, they gave me a lot of jabs. I didn't have enough shoulders and arms for all the vaccines I got. And they sent me to Africa. And for the first time, I arrived in a big city in Africa. This is Bamako in Mali. And it was one of the poorest countries in Africa at the time. And these are some of the first impressions I got. Okay? So in the city, there were lots of animals, which we don't usually see in European cities. At night, there were a lot of people sleeping on the roads. This was the nicest supermarket in the city. It was very, very dirty. The sewage system was open. People would sleep under these shacks. etc. The lab was also quite something. We had one room which was packed with stuff. There was nowhere we could work. I remember doing DNA extraction, we will see what it is, on top of a fridge because there was no space for me to work. Everybody was very nice. And then I was living in that hospital. There was an old guest house, and I would sleep in that hospital. And every morning, as I would step outside, there was a big number of people trying to get admitted in the hospital, right? And the conditions in that hospital was terrible. The government could just pay for some staff. The families had to pay for linen. They would camp outside to prepare food for the sick. They had to buy supplies and medications. So a lot of patients would die because they could not afford saline, they could not afford glucose, which are very inexpensive things. Okay? So terrible conditions. And I remember in a few days I was very happy waking up. I was always dissimulating my happiness because there were such terrible things happening outside there. And then something that really still stuck in my mind is that death was everywhere. The presence of death. In our European society, death is something really remote. In Africa, it was every day, everywhere, every day, it was there. The colleagues, for example, would bury people from malaria. You know, every week there was somebody going to a funeral. Okay? Every week there was a colleague that was sick at home of malaria. And the reason for that is that around 1998, the late 90s, malaria was so bad that in a country like Mali, where I was working, about a quarter of the children under 5 years were dying of malaria. Okay? We had on average 10,000 children dying per day, and a total of 2 million people dying per year of malaria. That was the situation towards the late 90s. So what did the world community do to answer that? Well, the World Health Organization led this project here called Rollback Malaria. It was a new program, not to eradicate malaria, but aiming to diminish malaria in Africa. And it was using a few new tools, okay? One invention was the long-lasting impregnated bed nets. So instead of having to retreat the bed nets every 6 months, the bed nets would supposedly stay active for 5 years now, which was a big advance. There was indoor residual spraying using pyrethroid compounds instead of DDT. There were better diagnostics to diagnose people sick with malaria. And there were also new drugs based on artemisinin. And there was so far no drug resistance to those drugs. So these are the main tools that are still used to this day and that have been actually quite effective against malaria. But 20 years later, we are still relying on that and awaiting some new developments. All right, so the reason for all these cases of malaria, this terrible situation, is this mosquito here, Anopheles gambiae. You see 2 females feeding here. It's a mosquito that really likes to feed on humans. It's very anthropophilic. Also likes to feed indoors and to rest indoors after the blood meal, which is actually our luck because it means we can kill them by spraying houses, and we can kill them with bed nets. It could be much worse if they were not having this behavior. But why was it so bad? Why were there so many mosquitoes? Well, it turns out that it's actually a species complex. In other words, a collection of different species. They look all alike morphologically, but they use different niches of the environments. So, for example, Anopheles gambiae, which we will talk about a lot more today, has a very large distribution. And then you have some species, Anopheles melas, for example, and merus, that use the coastal areas. And then you've got some smaller populations there. And I'm going to wear my glasses now because I need them. Sorry. Okay, there we are. So we have a total of 7 subspecies. And if you add up all those geographical distributions, you can see that they can exploit every ecologies in Africa, okay? Except for the Sahara and the cooler climate of southern Africa. Okay? So how did researchers identify them? Well, before the advent of molecular biology, they had to use microscopy, for example. And in mosquitoes, we're quite fortunate that some cells have very large chromosomes, chromosomes that we can look at using light microscopy. They're called polyethylene chromosomes. They're found in some cells that secrete a lot of proteins, and it's a solution for the cell to be able to produce this large amount of proteins. They duplicate the chromosomes so much that they become thick and visible. So researchers looking at those chromosomes realized that some of those populations of Anopheles gambiae had inversions in the chromosome. Part of the chromosome was inverted. And every individual in a given population had this inverted chromosome. Therefore, it's called a fixed inversions. And you could distinguish some populations with inversions, some populations with another inversion, etc. And then they tried to cross those potential species to check there was actually some reproductive barriers between them, because that's an objective criteria to identifying a species or not. So they combined this marker, which was these fixed inversions, with the capacity to generate hybrids. So they would take female, for example, cross the female with a male from the other population. They would check that the hybrid would be viable. They would also cross the hybrid back to one of the parental possible species. And if they could backcross, this is called backcrossing, if the backcross would be viable, then they would say, okay, there's no genetic incompatibility. These are not new species. But if they found genetic incompatibility, and they combine it with differences, fixed differences in inversions, then they could separate them as species. And that's how they identified those 7 species of the Anopheles gambiae complex. Because they cannot fully interbreed, they have so-called post-mating reproductive barriers. All right, so if we think in terms of the theory of speciation, there are different ways that these barriers can occur, these barriers between species. And if you look in a textbook about evolution, you will learn that isolation can occur in allopatry. And that happens when a single population is split in two, for example, by a mountain, by an ocean, leading to 2 populations. And over time, these populations become so different that they cannot interbreed anymore. And if they do interbreed, the hybrid is not adapted to either of the environment, so the hybrid doesn't live very well, and that's another form of reproductive isolation. And then you've got other scenarios where there's a bit less isolation. This is a split between 2 populations. That population is using a slightly different ecological niche, using the environment different, but it's not too far. But nevertheless, if there's not too much migration between the 2, you can have this same phenomenon and a split into 2 species. Another case is parapatric speciation, where the 2 populations are still in contact, and their speciation will occur provided that there's a strong selection for the 2 different environments and not too much migration between them. So these 3 conditions, we suspect, led to the 7 species, and this is perhaps more simply understandable because there has been a lot of variation in the climate over time. We know of glaciations in Europe. There are fluctuations in the climate between glaciations. And all of those fluctuations in Africa translated in variation in temperature and arid areas. And for example, this is the vegetation 20,000 years ago. And you see that the tropical forest, for example, which is suitable to some of those mosquito populations, is fragmented compared to now. And each of those fragments could potentially lead to a speciation event, if you think about the speciation modes we have seen. So fluctuations in climate can explain the formation of these different species of Anopheles gambiae. But what about this one? This is one of those species. It has a huge range still, Anopheles gambiae, ranging from the border of the Sahara, the sub-Saharan area, to very wet forests all the way down here to Madagascar. So people were suspecting that this must be further split. It's not possible, it's a single species, but the inversion trick and the crosses didn't work in this case. And for that, we had to wait for molecular biology to develop. And as some of you know, the genetic code of any living organism is encoded by nucleotides. It forms filaments. It's a double-stranded thing that coils on itself to form a DNA helix that itself coils to form nucleosomes. This is held together by some proteins called histones. And then these coils is supercoiled to form chromatin and chromosomes. Okay? So this is how the genetic information is encoded. These strings of nucleotides may be coding for genes. So you have a long list of genes in this code. Yeah? And by the late 80s, early 90s, it was becoming feasible to amplify this DNA by polymerase chain reaction. And we could then read some small pieces of DNA, of the information. This was called sequencing. Or we could just look at specific nucleotides, different places, and this was called genotyping, looking at some marker loci. All right? So this is what helped actually identify two populations within that bigger population we looked at just before. So Anopheles gambiae sensu stricto is actually divided in two populations. People looked at genetic differences in many parts of the genome, couldn't find any, until finally, on the X chromosome here, there was a difference. You know, some individuals were always of this genotype, some other individuals always of that genotype. This is in an area that is called ribosomal DNA. All right, so using those new molecular tools, they were able to define this new population, Anopheles coluzzii, which overlaps with Anopheles gambiae. Again, they look morphologically alike, but the difference with the species is that when you cross them, they can produce fertile offspring. So there are no reproductive barriers between Anopheles coluzzii and gambiae. Okay. All right, so we looked at that situation, and you will probably realize that for Anopheles gambiae and coluzzii, we are looking at this scenario of speciation, sympatric speciation. Sympatric speciation is a bit of a mystery still to this day, because it's a mode of speciation that requires really very narrow situation in terms of ecology and in terms of genetics. I'm going to explain that further. I was telling you that genetic exchanges between populations can slow down the rhythm of speciation, make it more difficult. And here I've symbolized one chromosome of individuals from this subpopulation, one chromosome from this population. When these two populations evolve separated by a mountain, they can differentiate genetically everywhere on the chromosome. No problem. If there are some migration because the populations are not too far from one another, you expect a bit of genetic exchange, and that kind of contaminates this kind of genetic divergence. In this case, you have even more gene flow. Okay? So speciation is slowed down, but imagine sympatric speciation. How is it that from within the same population, a group of individuals can evolve into something else? It's a big mystery. So there's been a lot of mathematical modeling, a lot of computer simulations to try to understand how the genetics could favor that. Right? So what needs to happen is that this subpopulation, they need to evolve a new way of exploiting the environment that gives them an advantage over other individuals, but they need to also evolve the capacity to recognize and to choose to mate with similar individuals that have this same capacity. And those genes of recognition, so-called assortative mating, have to stick together with the genes for ecological adaptation. Because if they're not close together, if there is a lot of interbreeding and still a lot of migration going on, those genes will be separated. Yeah? And there is no speciation possible. Therefore, you expect in this scenario of speciation areas where those genes are linked together and they are protected from crossing over, recombination, etc. These are the so-called islands of speciation. And for a long time, they were just a mathematical hypothesis, a theoretical hypothesis. Okay, so this is possibly what we would be witnessing with coluzzii and gambiae. All right. But going back to my first study in Bamako, we were interested because we had this new marker, the one difference between coluzzii and gambiae, to try to understand the mating system. I don't know if you can discern here. This is a cloud of mosquito. That is how mosquitoes breed. The males swarm at dusk, and the females visit the swarm. They pick a male, and then they fly away in copula. Right? So how is it that coluzzii and gambiae can separate and recognize one another? Is it the case that they recognize one another? Nobody knew at the time. It could also be that they mate randomly, and that somehow the hybrids that are viable in the lab die in the field. We didn't know. So the first study that we conducted, still one of my most cited studies, was using this novel technique in molecular biology, plus some forensic techniques used in human crime scene investigations. And I focused on the spermatica here in females. Females, when they mate, store the sperm of males in the spermatica for their lifetime. I managed to find a way to coagulate the sperm so that I could pick it and then conduct this DNA extraction to have the genetic information of the male, and then using the marker, understanding whether it's coluzzii and gambiae. And basically, this is what we found. When I dissected female Anopheles gambiae, the overall majority, 50 out of 51, had sperm of Anopheles gambiae males. Therefore, they can choose Anopheles gambiae amongst these clouds of mosquitoes. In females of Anopheles coluzzii, I found the same pattern. The overwhelming majority of females coluzzii had sperm from male coluzzii in the spermaticae. Only one mistake was found. Okay? We had a few cases where there was multiple mating, but they were rare. And interestingly, in that sample, we found one female that had a genotype compatible with the fact that she would be the progeny of a cross-mating. She would be a hybrid female. So the overall results is that assortative mating, the capacity of recognizing your own kind, is very strong in Anopheles. Yeah? It creates a very strong barrier between species. It allows these species to be maintained and not to fuse back again. But sometimes you find hybridization, so there's still a bit of gene flow. All right. So other teams use the same marker between coluzzii and gambiae to start discerning what types of ecologies they were using. And it turns out that Anopheles gambiae thrives in temporary rain pools. So in the season when the rain is abundant, they will mostly breed in those habitats. They will favor those habitats. They will outperform Anopheles coluzzii, and they will become very abundant. Anopheles coluzzii, on the other hand, seems to prefer more permanent waters. And it's been shown, other studies and colleagues from Africa showed that they're better able to avoid predators that live in these habitats. They tolerate more organic matter in the water that is typical of more permanent habitats like that. And this has been shown by transplant experiments, for example, moving the larvae, inverting the position of larvae, moving them across environments, and looking at their growth and emergence rates, for example. But what is very interesting here, and this is an idea I'm very keen on, is that some of those habitats have wild rice. And 5,000 years ago, in the Neolithic times, there were times in Africa where it was so dry, only the upper Niger River area in Mali had pockets of water where humans could sustain populations. And 5,000 years ago, they learned to domesticate wild rice. So human populations started surviving through these new skills, this new agricultural mode. And I believe that part of the success of Anopheles coluzzii stems from the fact that it evolved to use that new ecological niche. It was already pre-adapted to this kind of permanent habitats, and then it spread as the rice cultivation spread across West Africa. If it's true, then this would be one of those rare examples where speciation has been facilitated or even driven by human activities. And why do I believe that this may be the reason? is because the culture of rice cultivation is mostly limited to West Africa. If you go to Central Africa, there is much less rice culture. Those cultures don't culture rice. And then if you go to the south, people eat yam in those wetter areas. There is no rice cultures here. So you could explain the fact that coluzzii never spread beyond West Africa by the distribution of the main rice growing areas from a few thousand years ago. All right. But once we had this marker between gambiae and coluzzii, there are other aspects of their biology that we were interested in. And with molecular biology, you could look at other markers. And one marker that became very famous very quickly is the so-called KDR mutation. This is a change in the DNA that confers resistance to pesticides to mosquitoes that have the mutation. In other words, a mosquito that has that mutation is resistant to bed nets and resistant to pesticides, indoor residual spraying. So this is work I've done with my former boss, Greg Lanzaro, and a colleague from the Malaria Research Training Center in Mali, Sheikh Traoré. And we were very fortunate to have samples dating back from several years prior to the study from 4 villages in Mali where we had the two populations living together, Anopheles coluzzii and gambiae living together. And then we performed the so-called genotyping. And on this graph, this is the proportion of individuals that are fully resistant. In other words, they have the paternal and maternal version of the DNA carry the resistance, so they are super resistant. The gray ones have one of the copies, either paternal or maternal, that is resistant, and the white ones are susceptible. They are still killed by the pyrethroids. And you see that in each of the 4 populations, Anopheles coluzzii is fully susceptible, and it was for 11 years, from the first samples we had in the fridge to the last ones. From 1996 to 2002, no resistance whatsoever. 1993 to 2004, no resistance. Anopheles coluzzii stays susceptible. In contrast, all of the Anopheles gambiae populations have some level of pesticide resistance. A lot of times it's increasing over time. So now this is very crucial to vector control, okay? Because if you're in an area where you have such high levels of resistance in part of your mosquito population, you don't want to keep using pyrethroids. You should be switching to something else, right? Because you're losing efficacy, you're losing the battle against mosquitoes. Okay? But in terms of the speciation theory, what this told us is that there was no gene flow between those populations. Yeah? There was no hybridization, no backcrossing. Yeah? Because for years and years, no genetic exchange. You know, the coluzzii never received the resistance from Anopheles gambiae. So that meant very strong assortative mating, and somehow those hybrids were not surviving or something was happening. All right, so I'm going to change the topic a little bit and come back to speciation later on, but there's another type of intervention that would really benefit from understanding mosquito mating behavior. And this is a tool that we're hoping to deploy in the future. It's been deployed successfully in fruit flies. It's been deployed for a variety of insects. It's the sterile insect technique. And in Anopheles gambiae, unfortunately, it has not been successful because we don't understand the mating behavior very well. How does this technique work? You capture mosquitoes from your target population. You put it in the lab. You keep it, you colonize it, make it used to the lab, you grow it, and then depending on what system you're using, either you irradiate those mosquitoes using X-rays, or you use genetic engineering to sterilize them, and you create males that have sterile sperm. And you have to combine that with mass rearing to create thousands, millions of males. And when you release those sterile males, the hope is that those males will actually mate with wild females, okay? But you can imagine that in the process of colonizing, modifying them, growing them, a lot of things can go wrong. So I've been pushing since 10 years for more research in that field to optimize the sterile release for mosquitoes, and particularly for Anopheles gambiae, to have an additional tool to control mosquitoes and therefore malaria. All right, so there would be a lot of advantages in understanding how the females really recognize the males. Yeah? The exact mechanism, because we understand that, we can produce the perfect male for releases, but we can also produce better traps, for example, using the cues that female use. So in the steps during mating, and this is mating, that's what we suspect happens in swarms. The female and the males kind of come in contact at some point, and that's been based on photography. Then the legs touch, then the genitalia touch, and if everything goes well, they form a tandem and they fly away. Okay? But in those different steps, you could have mate recognition happening through a variety of mechanisms. So it could be that when females approach the swarms, they recognize the sounds, and they can distinguish males from Anopheles gambiae from coluzzii, for example. When the male chases the females, it could also be that the male recognizes the female that they need to mate with, and they use sound in that process. With leg contact and abdomen touching, there could be some chemical cues used for recognition. We don't know exactly. All right, so there are a variety of mechanisms that could really inform the production of males and also new traps. So despite not having so much funding for this type of work, we nevertheless went on with some of that, and this is work that's been done by some of the PhD students, Doug Patton, Esther Rekesheku, and Rohida Beshan. And I mentioned this work. I'm not going to explain that in details, but we look at the negative effects of selection and inbreeding in lab-produced Anopheles gambiae, and this is pioneering work that really put our lab in the forefront in this field of research. We also looked at the fitness costs of genetic engineering and radiosterilization in Anopheles gambiae, and this is with colleagues from Kiel, but also colleagues again from Mali, colleagues from Burkina Faso, and also the International Atomic Energy Agency supplied the X-rays. And then finally, we had a breakthrough. We put together a capacity building grant. This is one of the grants that were mentioned by Mark. And this was with colleagues in Mali, Sheikh Traoré again, Mamadou Koulibaly, and colleagues from Kiel, Hilary Hurd, and Paul Eggleston. And this was to be set in Mali. And I'm also mentioning this project because this is where I started to develop some other skills. We needed space, and we needed a secure insectary. And there was no money for an architect. There was no money for a building manager. So I said, well, let's just do it. And I drafted this, and I think my sister might recognize that because I asked her for some tips. She's an interior decorator by training. And this was my plan, 80 meters square of lab. And I spent 3 months altogether running between Kiel, my lectures here and Mali. And this is the space we received, and this is when, after a very difficult years, pushing the builders, this is the secure entrance. The doors came from Sweden. And then we have a molecular lab, and we have a large insectary. Okay? And this lab has been a success. It's one of the first facilities for functional studies in West Africa. And these days, it's used by a big consortium that is actually funded by the Bill and Melinda Gates Foundation. And actually, I'm employed partially on this project as the technical director. And so it's contributing to capacity building and collaborations with other projects. But in terms of at that time, what we were hoping to do, I was hoping to have a nice facility to do those mating studies. But that part of the budget was slashed, okay? So these were my makeshift cages, and we had to work in that. But we still managed, and Doug managed to conduct some experiments here. And in this type of experiments, we're trying to reproduce this condition where you have assortative mating. So we take coluzzii females here, and they look like that. The males have a bushy antennae. I don't know if you picked up the difference. So we've got these females, Anopheles coluzzii, and we take them when they're virgin, and we give them a choice between virgin males Anopheles coluzzii or virgin males Anopheles gambiae. And if they pick the right males, we know that assortative mating is working. If they don't pick the right males, we know that they don't choose assortatively. And in this case, we made the story more interesting, of course. So we had different experimental groups, and we used virgin females that were the progeny of wild mosquitoes, wild females, and we had virgin females that were the progeny of a colonized strain. And this is because we wanted to know if colonization influences the mate choice behavior, the sexual behavior of the adults. And then we had another layer, which was we were studying whether at the larval stage, whether those virgin females were reared in the lab or if they were reared outside. Did that affect the adult mating behavior? So we have those 4 experimental groups. These are virgin females, which are the progeny of field-caught females, and they were reared in the field. And on this axis, we are looking at whether they mate assortatively. So if they mate 50% of the time with Anopheles coluzzii males, it means that there is no mate choice. Basically, they cannot tell apart the males. And you can see these are 2 different measures of the same thing, but we're looking at the percentages. It's easier, and we can see that we had 80% assortative mating. Okay? It's not perfect, so we suspect that the cages may be a bit too small, maybe disturbing the swarming process a little bit. But overall, those females here that came from the field, reared in the field, are capable of choosing the right male. That's fine. The field ones that were reared as larvae in the lab did so too. They picked the right males. Interestingly, the ones that came from a colonized strain but were reared outside also mated assortatively. So despite the fact that they were colonized, being reared outside kind of protected them from not being able to choose. But the ones that were really bad were the ones that were from the colonized strain, reared outside, reared in the lab, sorry. These ones couldn't make the difference between the males. And this study, which actually is one of the very first studies focusing on Anopheles gambiae in those semi-field conditions, shows how complex mating behavior is as a trait, a mosquito trait. It really depends on the interaction between genetics and the environment, in this case, the rearing environment. And producing mosquitoes, when we are doing this and hoping that they will actually mate with our target populations, the intended population that we want to control, is really, really difficult. Okay. But on the positive side, I got more ambitious with my cages. I wanted to have my revenge. So on the next proposal, I proposed that to the Medical Research Council, and this is a grant that I got with my colleague Abdoulaye Diabaté in Burkina Faso now, because Mali started falling into civil war. And we set this construction in Bobo-Dioulasso. And I'm going to go quickly through that, but this is the end product. Again, one of the first such facility in West Africa, where we have a lot of compartments, where we can do some nice study of assortative mating. There is enough space to study the different aspects and mechanisms of assortative mating between Anopheles gambiae and coluzzii. In the meantime, my colleagues from Burkina Faso also made a big breakthrough. This is the mapping of the position of swarms in a village with very high mosquito density. This is VK7 in the rice fields. You can see the huge densities of mosquitoes. But importantly, they found that mosquitoes would swarm every time at the same place. Yeah? Predictably, days after day, you could go back and find the swarm at exactly the same position. Moreover, they found that the few swarms, this is all Anopheles coluzzii in that habitat. They like rice field, as I told you. There were a few swarms of Anopheles gambiae. They were also predictably always at the same place, but not the same place as Anopheles coluzzii. So therefore, there is so-called spatial segregation, which is another mechanism for keeping those two species apart. Okay. In Kiel, we had ambitious targets using new technology. So I told you that with molecular biology, we could sequence bits of sequences. If you can sequence all of the bits of the sequences in a chromosome, and you can assemble them using complex mathematical algorithms, you can reconstitute the whole genetic code of an organism, and that's called a genome. And based on genomics, you can do quite a few things. So we went for functional genomics, where we change the genome and we look at how it affects the mosquito. And there are other studies where you look at field populations and you compare the genomes of different field populations and you identify differences between them. And then there are also new methodologies where you can look at the level to which genes in the genome are expressed and translated into proteins, and the proteins are the constituents of living organisms. So some other research, some other groups, because we always have competitors, of course, did the comparative genomics, the first comparisons of the whole genome between Anopheles coluzzii and Anopheles gambiae. And here I've overlaid that over a map of the thick chromosomes. This is just to highlight the areas where they found that Anopheles coluzzii were different from Anopheles gambiae. So first conclusion is that there are no big differences between the two sister species in much of the genome. And you look at the third chromosome here, and they've got 3 chromosomes, chromosome 2, the right side, the left side, and the X chromosome, no noticeable difference, no consistent pattern to be seen, but they identified 3 regions. And surprise, surprise, it fitted the theory of islands of speciation. There were limited areas where those 2 sibling species were different. Okay. So from our perspective, since we are looking at genes of assortative mating and genes of ecological adaptation, we know that there must be somewhere here. The difference between coluzzii and gambiae must be hidden in those regions. So we had this smart idea. We decided, okay, we're going to in the lab, take Anopheles coluzzii, and we will replace this island by the island of Anopheles gambiae. And then we will see if it affects the assortative mating, if it changes the behavior and preference of females. And this is work done by Fred Abogaye-Antwi. It involved classic crosses using different genetic markers. And at the end of this process, here is a formal comparison of the dissimilarity between the genomes of our lines that have the coluzzii island on the X or the gambiae island on the X. And I'm asking you to look at the red line here. These are so-called recombinant lines. And you can see that here, the level of difference, which can be ranked from no difference to everything different. And you can see that our line, the red one, the difference between our mosquito lines with different islands of speciation is basically nothing on the 3rd chromosome as predicted, nothing noticeable on the 2nd chromosome as predicted, and then on the X chromosome it gets a bit messier, and this is where the island is, and we manage to have a large difference between our lines here. In other words, by using the full genome sequencing, we were able to confirm that our lines indeed were different just at this X island of speciation. So therefore, we're in a position to take those lines and do mate choice experiments. So we took females with one type of island and we gave them a choice between males with the 2 different types of islands. Everything else in the genome is identical. This is the result for the females. So the females with the coluzzii island picked males with coluzzii islands in 98% of the time. Okay? So they could really choose based on that one area of the DNA. Females with the gambiae island always picked males that had the matching island. Okay? So again, indicating that this is where those genes of recognition are hidden. In the lab test that we were using, males were not able to differentiate females. So there is no choice here. They picked 43% or 57% of one or the other, and we used males with the gambiae island, and they were unable to choose between females with the 2 types of island. So in our lab test, it's a female process of mate recognition, but it's very accurate in the conditions that we kept them in. All right, so in that process and using comparative genomics, we also identified putative genes that may be directly involved in the process of mate recognition. And we're doing further work, and the PhD students are doing further work on those genes. at the moment. But we were very keen, despite the fact that this was a clear indication, first, that the theory of speciation using islands of speciation was true, and also that the X island was probably the most important of the other islands. We were keen to use a field system to demonstrate the same thing, and also to look at the potential influence of those other islands. So another smart design, we decided to use additional markers within those islands. In other words, additional mutations that can distinguish coluzzii from gambiae inside those 3 islands. These are markers within the X island, these are markers within the 2L island and the 3L island. And you can genotype individuals that you capture in the different swarms, and you can describe them in terms of whether they look like pure Anopheles gambiae, and these are the nucleotides that are typical of Anopheles gambiae, a pure Anopheles gambiae here in dark blue. And then we have individuals that are pure Anopheles coluzzii in light blue. And then, of course, we picked a population where we knew there was interbreeding, recent interbreeding. So we had actually a lot of hybrids in that population, in yellow. You know, they share part of the genotype of gambiae and part of the coluzzii, 1 allele from each. And then there are some other individuals that match back crosses, a hybrid that has mated back with one of the parental adults. And these have mixed genotypes. You can see they have a bit, this one has a bit of coluzzii within a lot of gambiae genetic material. This one has the 2L that is coluzzii-like, etc. So we can do that for a lot of individuals, and then we generate these maps that summarize populations. So in the population that is in the rice fields, you know that it's mostly coluzzii, so the population is mostly blue, and we have quite a number of males that are pure coluzzii. All of those markers here are light blue, pure coluzzii. And then we realize, wow, there are coluzzii on the X, but a lot of times in the 2L island, they are hybrids, or they are even looking like a gambiae. And on the third island, the one on the 3rd chromosome, they were mostly coluzzii. So we got a lot of individuals like that. And over time, the proportion of individuals that are pure coluzzii were decreasing. In other words, the whole population was turning into a hybrid population. And most of the coluzzii individuals actually borrowed the island of speciation from the Anopheles gambiae. Okay? Why? We could not understand. But the important thing is that we had a variety of recombinants with bits and pieces from the 2 species. In females, we found grossly the same. In Somoso, another village where we actually had a lot of gambiae and few coluzzii, the reverse situation, you see that we have a lot of pure gambiae. Yeah? There's nothing that surprising happening here. This is what you would expect, except that you have a few pure coluzzii here, and then these pure coluzzii over time turn into hybrids, just like we observed in this population. And then within gambiae, there are quite a few individuals that have a mixed 3rd island. Yeah? The 3rd island is hybrid. Okay? So this population seems to be very tolerant of having a foreign 2L island, and this population seems quite tolerant of having 3L foreign island from coluzzii. Of course, we were interested to try to associate one or the other island with whether these individuals were found in coluzzii swarms or gambiae swarm. This was the trick. Okay? And in this table, we summarized the degree of association between those 3 islands and whether they swarm in a coluzzii swarm or a gambiae swarm. And if you look at the X chromosome, you find that 100% of those individuals have an X island of coluzzii. Yeah? So the best match in terms of going to a certain type of swarm seems to be the X for coluzzii. For gambiae, nearly the same, 99.7% of individuals found in a given type of swarm has a matching X. On the 2L, it's totally broken down, no more correlation. in Anopheles coluzzii. Quite a strong correlation in gambiae. And on the 3L island, you have a very strong association, but not as big as on the X. And in gambiae, the association is not as strong. So overall, what this tells us is that the X island is again the one that's determinant for assortative mating. And this time it's a different phenomenon. It's not choosing amongst virgin adults in a small cage. It's what happens in the field in terms of swarming. It's male-driven. It's not female-driven this time. So we found two independent mechanisms of assortative mating associated with an island of speciation. And therefore, it's strong support towards this theory of speciation islands. All right, so how is it that this island on the second chromosome has been taken by coluzzii? What advantage does it confer them? Well, you remember this name, KDR. This is the mutation that confers resistance to pesticide. This is the mutation that was never found in coluzzii before. It turns out that it is inside the island of speciation of the second chromosome. Therefore, the reason why we find increasing numbers of islands of speciation gambiae into coluzzii is because they are taking the KDR resistance from gambiae through hybridization. And here we follow the KDR marker in those populations, and you can see that in Anopheles gambiae, all individuals are resistant. This is in dark red. And then in 2006, there were some coluzzii that were susceptible, but quickly they were all hybrids. Yeah? And this is what we observed when we looked at the islands of speciation. In Sumuso, it happened a bit later, but coluzzii became hybrid. They took the island of speciation and the KDR locus and integrated them inside their genome, and they are now all fully resistant. In other words, we came from a situation where only gambiae was resistant, and within 6 years, we have coluzzii that is also completely resistant because of this hybridization. Okay? So why is that so? Well, paradoxally, it's probably because we have more and more bed nets. The selection pressures that we are imposing on populations is increasing because the proportion of people that sleep under bed net is strongly increasing. So this is in 2005. Average proportion of people on the bed net was very low, 10%. If you look now in 2013, you can see that the proportion of people under a bed net is much higher. The benefits of that, which is the effect of rollback malaria, is that the number of cases of malaria have dropped drastically in 9 years. Yeah? But on the negative side, we are now pushing mosquitoes very hard with selection pressures, and we have increasing pesticide resistance in mosquito population. And this kind of shows the hotbeds of pesticide resistance in Africa. There is pesticide resistance in many, many places, but notice West Africa. Yeah? West Africa is resistance everywhere. So is there something particular with rice fields and West Africa and Anopheles coluzzii that generates these highly resistant populations? Okay, so this is one of the last studies we made very recently, and we went for this habitat here, which is rice field dominated. This is VK7. You've seen that name before. And this is Somuso. This is the different ecology. It's dominated by Anopheles gambiae, and it has a few coluzzii. And we were trying to understand what are the causes of hybridization, because hybrids don't occur that frequently. When do they occur? What causes those conditions where hybridization occurs? And this is the summary. This is, again, work done by Nang, which is a PhD and then a postdoc on the big project. And he went to the rice field area, and what he did, he stood by swarms at dusk, and whenever a couple left the swarm, he would snatch it. Okay? This is a huge amount of work. He didn't do that alone. He had some helpers. But this work captured a total of, I don't know how many couples in copula. We have over 200. It's an amazing amount of work. It looks like basic ecology, but it's terrific work. And what he found is basically in that area of the rice fields where you have huge densities of mosquitoes dominated by coluzzii with a few gambiae, he found some interspecies pairs. So there were 4 females of Anopheles gambiae paired with Anopheles coluzzii males. Yeah? The majority were paired within species. Yeah? They mated with the right males, but you had, a certain percentage of cross-mating. If you look at the Somuso setting, where Anopheles gambiae is dominating and coluzzii is rare, there were no interspecific mating. And the reason may be that the overall density is quite low. Yeah. So we think that what is happening at certain time of the year, Anopheles gambiae females and males are so rare and they are lost among so many Anopheles coluzzii that they cannot find their own swarms. Yeah? They try different swarms, they don't find the right one, they are lost in this high density of mosquitoes, and therefore, after a while, they have to choose. Either they cross-mate or they give up on mating altogether. So they do the best of a bad job strategy and they cross-mate. And this could explain why, in rice field, at certain times of the year, you get hybridization. So the combination of this high density and conditioned rice fields with the selection pressure of the bed nets creates these special conditions where suddenly those 2 species are cross-talking, exchanging genetic material again. Yeah? So paradoxically, rice cultivation 5,000 years ago led to the new species. And now 5,000 years later, because of the added selection of bed nets, we see those species coming back together and exchanging increasing genetic material. Okay? Now, in the context of food security in Africa, this is very important because typically West Africa was the big area growing rice. And this is the growth in hectares of rice fields in Africa. All of the countries are investing immensely in rice cultivation, yeah, because growing populations need a lot of rice. It's a very good food, very rich food. So West Africa is continuing to lead in terms of culture of rice field. But worryingly, East Africa is also picking up. Okay? And East Africa, remember, there's no coluzzii in East Africa. So now we're creating new niches for coluzzii. Therefore, there will be a lot of habitats where coluzzii can settle. And moreover, you can have creation of populations. These hybrid population, and here this is something that came a few months back, super mosquito resistant to insecticide-treated nets emerge in West Africa. Well, this type of scenario, in my view, is going to happen more and more. We will have those hybrid population of super mosquitoes resistant to about everything. Yeah? And it's going to be increasingly so as we bombard them with more and more pesticides. So understandably, I'm very, very interested in the management of vectors in those areas and balancing food sustainability, food security, and vector control. With that, I'm approaching the end of my talk. There were lots of people involved in this selection of work, of course, and I've interacted with a lot of people from the Center of Applied Entomology and Parasitology, but amongst the staff in the labs, these are some of the students, master students, PhD students that contributed to this work. I collaborate a lot with this institution, Institut de Recherche des Sciences de la Santé in Burkina Faso. I've got a very good collaboration with them. And again, we had PhD students on those grounds, postdocs. We worked a lot in Mali, and again, we have a number of postdocs and staff who contributed to this work. And for genomics, I tend to work with the Liverpool Center for Genomic Research, and they're always very helpful. All right, so over the years, I've received money for quite a few foundations and other organizations, some governmental ones. But I have to thank some other people too, because I was raised in a family that were very tolerant of my extravagancies, my naturalist tendencies, and they really nurtured that, they encouraged that. And thanks to them, I'm the naturalist that I'm now, and it really helped me throughout my career. So this will remind them of a few anecdotes and a few adventures, a few notorious escapes, for example. I'm also very thankful to having great supervisors. This is Professor Heinz Richner, a behavioral ecologist at the University of Bern. He just retired last year and he's always been very, very supportive. And then Greg Lazzaro is very, very famous in population genetics of Anopheles gambiae, the Anopheles gambiae complex. And then finally, I'm very, very fortunate that I have terrific colleagues and collaborators in Africa. Okay? These are people that are my friends because we work so much, I don't have so much social life. And they're really inspiring, they're really driven to develop research in Africa, develop strong research, strong malaria control. They want the same standards that are found elsewhere in the world. And they're an ever source of inspiration and drive in my work. And then finally, I'm very thankful to have found a research center that kind of understands my mode of functioning and they understand those projects abroad and the demands of these international collaborations. Thank you very much for your attention.